What is the human microbiome? The human microbiome is the collection of microorganisms that live on and inside the human body. These microorganisms include bacteria, fungi, viruses, archaea, and other microscopic organisms. They live in many different areas of the body, including the skin, mouth, nose, digestive tract, and urogenital system. The term microbiota usually refers to the microorganisms themselves, while microbiome is a broader term that can include the microorganisms, their genes, and the biological activities they carry out. The human body does not have just one microbiome. Different parts of the body contain completely different microbial communities because each area provides a different environment. The microorganisms that thrive on oily skin, for example, are different from those that live in the mouth or the large intestine. These microbial communities are not simply passengers. Many of them interact continuously with human cells and can influence digestion, metabolism, immune function, and protection against potentially harmful microorganisms. How the microbiome develops. The human microbiome begins developing very early in life and continues to change throughout childhood, adulthood, and aging. Its composition can be influenced by many factors, including diet, environment, medications, infections, lifestyle, and age. This means the microbiome is not a fixed collection of microorganisms. It is a constantly changing ecosystem. Even within the same person, microbial communities can change over time as eating habits, medications, health, and environmental exposures change. The oral microbiome. The mouth contains one of the most diverse microbial communities in the human body. Microorganisms live on the tongue, teeth, gums, inner cheeks, and in saliva. Different surfaces inside the mouth create different environments. The surface of a tooth, for example, provides a hard structure where microbial communities can form dental plaque. Some oral bacteria metabolize carbohydrates and sugars from food and produce acid. When acid production becomes excessive and frequent, it can damage tooth enamel and contribute to cavities. Changes in the oral microbial community can also be associated with gum inflammation and periodontal disease. The skin microbiome. The skin also supports large communities of microorganisms. They live on the skin's surface and around structures such as hair follicles and sebaceous glands. However, different areas of skin support different microbial populations. Oily areas such as the face and scalp provide a very different environment from dry areas such as the forearm. Moist areas, such as the armpits, create another type of habitat. These microorganisms interact with the skin barrier and the immune system and can compete with other organisms attempting to colonize the skin. The nasal and respiratory microbiome. Microbial communities also exist in the nose and respiratory tract. The nose is constantly exposed to microorganisms from the surrounding environment through the air we breathe. Mucus, immune defenses, temperature, moisture, and oxygen levels all help determine which microorganisms can survive in different regions. Microbial density varies greatly throughout the respiratory system, so it should not be thought of as containing the same enormous microbial populations found in the large intestine. The gut microbiome. The digestive tract contains some of the most extensively studied microbial communities in the human body. The stomach, small intestine, and large intestine all contain microorganisms, but the largest and most complex populations are found in the colon. One of the most important functions of gut microorganisms involves processing parts of food that human digestive enzymes cannot completely break down. Dietary fiber is an important example. Certain gut bacteria can ferment fiber and produce compounds known as short-chain fatty acids. These include acetate, propionate, and butyrate. These microbial metabolites can interact with intestinal cells and participate in processes involving energy metabolism and immune signaling. The gut microbiome also helps create what scientists call colonization resistance. The intestine contains limited space and nutrients. When established microorganisms occupy these ecological niches, they can make it more difficult for some potentially harmful microorganisms to become dominant. This microbial ecosystem can be disrupted by major changes such as illness, diet, or certain medications. Antibiotics are particularly important. Antibiotics may be essential for treating bacterial infections, but they can also affect bacteria that normally live in the intestine. When parts of the normal microbial community are removed, ecological spaces and nutrients may become available to other organisms. One well-known example involves Clostridioides difficile, or C. difficile, which can sometimes grow excessively after antibiotic treatment and cause severe intestinal disease. The microbiome and the immune system. The immune system is in constant contact with microorganisms, particularly at body surfaces such as the intestine, skin, mouth, and respiratory tract. Immune cells must perform a complicated balancing act. They need to respond to dangerous pathogens while tolerating many microorganisms that normally live in or on the body. The relationship is therefore much more complicated than simply dividing microbes into good bacteria and bad bacteria. The effects of a microorganism may depend on its species, location, abundance, and the condition of the person hosting it. An organism that normally exists harmlessly in one part of the body may cause disease if it enters another location. The gut-brain axis. The digestive system and the brain communicate through several biological pathways, collectively known as the gut-brain axis. Communication can involve the nervous system, immune signals, hormones, and substances produced by microorganisms. The vagus nerve is one important communication pathway between the digestive tract and the brain. Researchers are actively studying how changes in the gut microbiome may influence this communication. However, the gut microbiome should not be described as directly controlling thoughts, personality, or emotion. This is an active area of scientific research, and many of the relationships are still being investigated. The urogenital microbiome. The urinary and reproductive systems also contain distinct microbial communities. The vaginal microbiome is particularly well studied. Its microbial composition can influence local acidity and can interact with the tissues and immune defenses of the reproductive tract. Once again, the important principle is that every body location creates its own ecological environment. A microorganism that is normal in one area may not be normal in another. What is dysbiosis? The word dysbiosis describes a disruption or alteration in a microbial community that may be associated with impaired function or disease. This might involve the loss of certain microorganisms, excessive growth of others, or changes in overall microbial diversity and activity. Scientists have identified microbiome differences in people with many different diseases. However, an important distinction must be made. Finding a different microbiome in someone with a disease does not automatically mean that the microbiome caused the disease. The disease itself, medications, changes in diet, inflammation, and other factors may also change the microbiome. What changes the human microbiome? Many factors can influence microbial communities throughout the body. Diet is one of the most important influences on the gut microbiome. Medications, especially antibiotics, can also produce major changes. Other influences include aging, infections, environmental exposure, sleep patterns, stress, physical activity, and long-term lifestyle habits. Because so many factors are involved, there is no single microbiome composition that can be considered perfect for every person. Supporting a healthy gut microbiome. One of the most practical ways to support the gut microbiome is through a varied diet containing fiber-rich food. Vegetables, fruits, beans, whole grains, nuts, and seeds provide different types of carbohydrates that intestinal microorganisms can use. Some fermented foods, including yogurt, kefir, sauerkraut, and kimchi, may also contain living microorganisms. However, not every fermented food should automatically be considered a probiotic food. Prebiotics are substances that are selectively used by certain microorganisms and can provide a health benefit. They are essentially nutrients that support particular microbial populations. Probiotics, in contrast, are live microorganisms that are intended to provide a health benefit when consumed in adequate amounts. But probiotics are not interchangeable. Different species and strains may have completely different effects, and evidence varies depending on the health condition and the specific product. Antibiotics should also be used appropriately rather than avoided completely. They can save lives when bacterial infections require treatment, but unnecessary antibiotic use can disrupt microbial communities and contribute to antibiotic resistance. Conclusion. The human body is not simply a collection of human cells. It is also home to complex microbial ecosystems. Microorganisms live on the skin, inside the mouth, throughout the digestive tract, and in several other areas of the body. Some help process food. Some interact with the immune system. Some produce biologically active substances. And many compete with potentially harmful organisms for space and nutrients. The microbiome is constantly changing, and scientists are still discovering how these microscopic communities influence human health. Understanding the microbiome does not mean trying to eliminate bacteria or searching for one perfect collection of microbes. It means recognizing that human health involves an ongoing relationship between our own cells and the enormous microbial world living alongside us.